Welcome to the Cruise Biology. In this video tutorial, we are going to talk about structure of DNA and RNA. So before we are going to discuss about the structure of DNA and RNA, we have to understood the what is nucleic acid. So the first of all, what is nucleic acid? Nucleic acid, there is the complex organic substance that is present inside the living cell. So there is the two types of nucleic acid, that is DNA and RNA. Nucleic acid is a complex organic substance that is present inside the living cell. So the importance of this nucleic acid, that is it contains the heredity information that is passed from the one generation to the another generation. This heredity information that is from your parents, it is passed to the offspring, that is your child, right? So that is the nucleic acid. That is the most important organic substance present inside the living cell. So there is the nucleic acid is made up of the monomer subunits. Now these monomer subunits are the nucleotides. Either in DNA or RNA there is different nucleotides, and these nucleotides is contains the three components. First one, phosphate group, sugar molecule, and there is different bases. We are going to discuss all the things in detail, which are the types of sugar, types of base, what is the components in DNA, RNA, in the nucleic acid. So the nucleic acid is the polymer of nucleotides. That is the nucleic acid, and there is two types of nucleic acid, right? And there is three components of nucleotide. So we are going to discuss in details. So DNA and RNA is made up of nucleotides, and this monomer of nucleic acid, that is nucleotides, and the polymer of these nucleotides, that is the DNA or RNA. That is the two types of nucleic acid. So now we are going to discuss about the structure of DNA and RNA, right? And there is arrangement of nucleotides, that is called polynucleotides. Now the first of all, there is the phosphate group and the sugar molecule is attached, and it makes the sugar phosphate backbone. Sugar phosphate, and it makes the backbone like structure in the arrangement of nucleotide. So backbone, that is sugar phosphate backbone. Now different type of bases that attach with this sugar molecule. At every sugar molecule there is different type of bases is present. In the next we are going to discuss about which type of sugar in the DNA or RNA, which type of base in the DNA and RNA, and how phosphate and sugar molecule is attached. And if I talk about one nucleotide, that is the phosphate, sugar and a base, and the second nucleotide that contains phosphate, sugar and base. So there is attachment of one nucleotide to the next nucleotide, and it makes the polynucleotides. So if I have to discuss, I have to draw the structure of first sugar phosphate backbone. So there is different type of sugar, that is pentose sugar, that contains the 5 carbon present. In the RNA there is the this sugar is cyclic in nature, and in the RNA there is the ribose type of sugar is present. Ribose type of sugar in the RNA, and in the DNA there is deoxyribose sugar is present. Deoxyribose, that is present in the DNA. That is about sugar molecules. Now if I have to draw this sugar, this base is attached with the sugar molecules. Every sugar molecules there is attachment of base. So there is different type of bases present in the DNA and RNA. First, in the DNA there is 4 type of bases, that is A, G, C and T. So A stands for adenine, G stands for guanine, C stands for cytosine, and T stands for thymine. That is in DNA. But in RNA there is also 4 type of bases. The difference is, instead of this thymine there is uracil is present, that is U. So A, G, C and uracil instead of thymine. There is little bit difference in the structure of thymine and uracil, that is only the methyl group is attached. In the there is structure is different, but there is similarity between thymine and uracil. Now if I have to classify this base, so there is 2 purines. It contains of 2 rings. So the common purines, that is adenine and guanine, that is called purines, and pyrimidines, that is contains one ring structure, that is cytosine, thymine and uracil, that is the pyrimidines, and purines, that is adenine and guanine. Now pyrimidines, cytosine, thymine and uracil, there is difference in the ring structure. There is 2 ring structure and there is 1 ring structure is present. Now if I have to draw all these structures, so the first one I have to draw the sugar molecules. So that is the 5 carbon sugar that contains the 5 carbons, and from this 5 carbon, how we can calculate from the first carbon to the 5th carbon is attached there. So here is the first carbon, here is the second, here is the third carbon, here is the 4th carbon, and the 5th carbon is here. 5th carbon is here. That is the structure of sugar molecule that contains the 5 carbon. Now I have to draw the number of carbon. So there is the arrangement of carbon: first, second, third, fourth and 5. That is the prime, first prime and second prime, third prime. So that is sugar. Now base is attached with the sugar. So at the first carbon base is attached, right, at the first carbon, and at the 5th carbon of sugar there is attachment of phosphate group, the ion of phosphoric acid. I have to draw the structure of phosphate group. Phosphate, oxygen is attached here, oxygen, right. That is the phosphate group is attached here, and here the base is attached with the 5th carbon. I can draw that. So that is one nucleotide. Here is the one nucleotide. Now the second nucleotide is added with the 3rd carbon of sugar, and in the DNA and RNA there is difference in the sugar. First I talk about ribose sugar and deoxyribose sugar. So there is presence of OH group in the sugar molecule, that is ribose sugar, and there is lack of OH group in the sugar. So the presence of this OH group in the sugar, that is, it makes the difference, and here that is the at the 3rd carbon it is attached, it is free group. To to react with the another nucleotide. There is attached a free OH group, and here the free phosphate group that is attached with the 3' end group. Here is the sugar base and phosphate sugar base bond is there, and every sugar, here is the phosphate, right? So I can write this phosphate, here is the sugar, right, and it makes the sugar phosphate backbone, and here is the base. Okay, right, here is the base. So this is the structure of DNA in the RNA. Now in the RNA, I talk about the RNA. There is difference in the sugar. There is presence of OH group and there is lack of OH group. That makes the difference in DNA and RNA. Now due to this OH group, the RNA makes the more reactive than DNA, and it is susceptible to hydrolysis. So at the second carbon in the RNA, there is OH group is present, so that is ribose sugar. It makes them more reactive than DNA, right? It is make more reactive than DNA, and it makes susceptible to hydrolysis, right? So that is the structure of nucleotide. Now every nucleotide is attached with the phosphodiester bond. So these sugar molecules, if I have to draw the one nucleotide that contains the 3 components, the sugar, base and phosphate group. So every nucleotide is attached with the phosphodiester linkage. So at the 3' end of one nucleotide, there is attachment of the 5' phosphate group. So there is only oxygen is here, is attached with the phosphate group. At the free OH group, oxygen is reacts with the phosphate, and this oxygen is the 5th carbon. This is the next nucleotide. Base is attached here, right? There is the 3' end. In the 1' end, there is the numbering of carbon 1, 2nd, 3rd, and here is the attachment of next nucleotide at the 3' carbon, and is the pentose sugar at the 3' carbon. There is attachment of the next nucleotide, and this makes the attachment. This is the nucleotide, first nucleotide. There is and here is the 5th carbon of the one sugar. Here is the sugar and base at first carbon. Now at the 5 carbon sugar, at the 5 carbon and at the 3 carbon, there is the different groups, free groups, and 2 nucleotides joins with this phosphodiester linkage. Phosphodiester linkage. These 2 nucleotides is joined together by this linkage, that is phosphodiester linkage. Now at the 5 carbon and the 3 carbon of the nucleotides, there is groups that is free to react with the another nucleotide. So at the 5' end and here is the 3' end, there is groups that is free to react with the another nucleotides. So at the 5' end and at the 3' end, there is OH group at the 3' end, and at the 5' end there is free groups to react with the nucleotide. So which group? That is at the 5' end, free phosphate group is reacts with the another nucleotide, and at the 3' end there is free hydroxyl group, free OH group is present to react with the next nucleotide. So if I have to write from the 5' end to the 3' end, so I can write the different bases for the naming from the 5' end to 3' end. So if I have to write the naming, if I have to draw the naming, so I can write from the 5' end to 3' end, different bases that I have to write. So the 5' end, suppose in the DNA, if I have to write the DNA, so there is different adenine, guanine, thymine, adenine is I can write. So you can write the nucleotide. That is all about the structure of DNA and RNA. I hope this is helpful to you guys. Thanks for watching, and like, subscribe and share my YouTube channel.